Studies, the IAS Third. I am the co-director for science there, and on behalf of my colleagues, it's a pleasure to see you all coming along to one of our lectures this evening in Thurrock University. So, the IAS, for those who aren't familiar with it, is an interdisciplinary think tank, which brings together people to address projects which are very broadly defined. So, an excellent exemplar is the one we're going to visit tonight, which is antibacterial clay materials. And this brings together both people looking at the physical sciences of how clays, minerals might be antibacterial, how the biology is impacted by the clay mineral, but also anthropologists to study how people use these materials. And in a wider context, projects may also involve artists, for example, or social scientists, all coming together to address one theme. Our speaker tonight is Professor Hailiang, who is it's quite a long title, Professor of Geomicrobiology, Geochemistry and Mineralogy at Miami University. So confusingly, it's not in Florida, it's in Oxford. And confusingly, that Oxford isn't in Oxfordshire. If you've kept up with me so far. Professor Dong obtained his PhD at the University of Michigan and then pursued postdoctoral studies at Princeton University before becoming assistant professor, associate professor and professor by 2009. He's been a program director of the Geobiology and Low Temperature Geochemistry program at the National Science Foundation, the primary US funding body. And he has also been Geoscience Director, won numerous awards. He's been Miami University Distinguished Scholar, the Marianne L. and Christy M. Jackson Mid-Career Clay Scientist Award, and also National Science Foundation Public Service Recognition Award, as well as the Geological Society of America Fellow. He is editor-in-chief of the journal Chemical Geology and associate editor of Geochemica et Phosphochimica Acta, Clay and Clay Minerals, and Geomicrobiology Journal. He has published over 230 research papers, a very prolific scientist over his career in various fields, including clay mineralogy, low temperature geochemistry, geomicrobiology, environmental microbiology, and environmental engineering. His research is focused on four broad areas: mineral microbe interactions, the remediation of heavy metals and radionuclides, as well as microbial life in extreme environments such as the deep subsurface or saline lakes, and microbial response to climate change. So, without further ado, I give you Professor.
All right, thanks very much, Chris, for nice and lengthy introduction, and thanks very much for coming here. And I know it's a rainy day, and also this week is a little special, right? And a lot of people are going on strike. So I give, I guess, the first similar talk, in our group, and this is almost the second last public lecture, and so I want to just expand on what I talked about before, and into a little broader area. And this is supposed to be a public lecture, so I want to make this a little more public. And so, yes, when I first interviewed at Miami University, actually I was confused myself, right? And Miami University, I said both Oxford and Miami are great names, and I don't know if there's a connection, my Oxford versus Oxford here, but definitely there's a connection between this Miami here in Ohio and Miami in Florida. This is named after an Indian tribe. So even today during our celebration and big celebrations, and so we always invite chief of the Indian tribe back to campus, even to today. And so now the tribe lives in Oklahoma, but they migrate from Ohio to Florida to Oklahoma, and via very long journeys. And so the region that's here, right here, in the southwestern Ohio, entire region is called Miami. So we have Miami River, Miami Valley, Great Miami River, Small Miami River, and lots of Miami, and lots of names named after Miami. And so this is our campus. This is my building right here, and this is our logo. So this building is almost 200 years old, but it looks fairly new because it's newly renovated. And then a lot of this work I presenting tonight also is done by students and postdocs and collaborators in China University of Geosciences. And this name is also confusing because there are two universities under the same name. One is in Beijing, and one is in Wuhan. It's about 2 hours flight to the south of Beijing. And both came from the same university. It's called Beijing College of Geology. And during Cultural Revolution, the main campus was moved to Wuhan. Actually, several places in Hubei province. But some people did not want to move and remained on the same campus. But a large fraction of campus was occupied by various other agencies and companies. So now there's only about half of the original campus remains. And so for a long time, when I was doing undergrad there, the main campus, the undergrad students were mostly in Wuhan campus, and the one in Beijing was mostly graduate school. So it's like long travel, always back and forth, and my professors always travel and always ask us to buy tickets to go from almost like from here to Newcastle every day. And but this is like 2 hours flight, not like a very short trip by train. So, but anyhow, so now these are two more or less independent universities. There's still a lot of connections, a lot of state key laboratories are joint, sponsored by two universities. So today I want to basically talk about this general big area of medical geology. So, and the clays fit into this medical geology and medical mineralogy. It's just really, it's crossover between chemistry, materials, geochemistry, mineralogy, and obviously life sciences and biology. So this kind of interdisciplinary study requires expertise from multiple fields and multiple disciplines. So actually I want to give a little historical perspective as to why there's such thing as medical geology. And as geologists, we have a lot of applications in medical field. Actually, interesting history. So it goes back to hundreds of years ago. Actually, the fathers of geology as a discipline, most of those are MDs. For example, this is a very famous geologist, and we use his principle in our textbook, Nicholas Steno, and he studied medicine at a university in Denmark and Netherlands in 1664. And his specialty was in circulatory system, including heart, because of blood circulation. And he worked in a hospital as a physician, and but he is also a priest in a Catholic church. But a lot of fundamental laws in geology, for example, stratigraphy and paleontology, discovered by him. And so a simple example is the law of superposition. So if we deposit sediments and later on lithify into sediments, always to go from bottom up. So the ones on the up are always younger in age than the ones below. Right? Today it seems like very simple, but back then this was undiscovered, and he discovered all that. And then a whole bunch of other stuff. We have, say, cross-cutting relationships. If one rock is cutting into another rock, so the cutting rock is younger than the one that's being cut. So this gives us relative age between different rock types, because back then there's no way to determine the age of rock in an absolute sense, only relative. So he worked out all these relationships. So clearly geology and medicine actually came from the same people. And James Hutton is another very famous guy here, and he got his MD in Netherlands, and his PhD dissertation was on circulation of blood. But he found it's the most phenomenal law in geology, and basically it says all the natural laws and processes that operate today are the same as the ones in the past. So by studying the processes in modern day, we can infer what happened in the past. Okay. So again, this seems to be very simple to us today, but back then, this is actually a major discovery, and this is a classical law of geology in textbook. And during my freshman year, I studied all these things. And then, interestingly here, the oldest geological society in the world, that's Geological Society of London. It was founded in 1807. And out of 13 founding members, 4 of these are medical doctors. So they had a strong interest as a hobby, right, in geology. And so this is a physician, but he is also a mineralogist. And he had the idea to form a geological society and served as the first president of the society. Then followed by several others. They are physicians, and James Laird served as secretary, and for other things over here. But and it is supposed to be 4, and I'm going to see the British Museum, the Natural History Museum, to see if I can find these people. But this, by the way, pictures from a book. And so clearly, if we trace back geology and medicine, right, back to the roots, they actually came from same roots. Okay, it's just these trees of knowledge have grown apart over time into separate disciplines. Now, in geology, geologists are perceived as holding this hammer and look at rocks, right, and go to mountains, look at plate tectonics, volcanoes, and earthquakes. As medical doctors, they use this, right? You guys know what this is, to hear your heart beating and to see what's wrong in human body. And so now, really, over time, these 2 disciplines have separate further and further away from each other. Now today, in my lecture here, I hope you get a sense that these 2 things are coming together again in this field of antibacterial clays and beyond. And so, for example, right, bacteria, for example, they don't care, right, what environment, maybe inside the human body or maybe in rocks. Okay, they don't sense and they don't define what a geology of medicine is. They just want to make a living there. Okay, and there's people, there's us, who really defines geology and medicine using different tools, looking at different objects, different things, and conclude different things. So today I want to give you guys a sense of what a medical geology is. These 2 old disciplines are actually coming together again. And as a geologist, we can study a lot of medical applications of minerals, vice versa, right? Medicals care about what kind of, like, for example, somebody has a kidney stone, okay? This is clearly a medical problem. But that kidney stone, right, actually we use a lot of geological tools to identify what kidney stone is, and the structure and the chemistry of that kidney stone, and why somebody has kidney stone. The reason for that, then hopefully to find some way to treat. So in our body, bones and teeth, we have all kinds of minerals. Okay, obviously as geologists we study minerals, but these minerals occur in a lot of our body. And in our teeth, for example, who can say what's in our teeth? Like what mineral? Anybody? Calcium. Calcium what? Just calcium? Calcium is not mineral, it's not solid. Calcium carbonate, calcite, more like a calcium phosphate, peptide. Because of calcium carbonate easily dissolves. Now we have a lot of acidity in our body, so cannot really stay for very long. But calcium phosphate, we call peptide, is very insoluble. Okay, and kidney stone, for example, is usually calcium oxalate and some other organic mineral we call, right? It has very low solubility. It does not dissolve in acidic environment. So, and then if some people have surgery, right, and then bones I could go back up, right, to rejoin, a lot of mineral precipitation in these processes are really the same as what we observe in nature. Right, just different environments. And so, for example here, calcification in cardiovascular veins. And this is a publication from one of my colleagues. And so he observed all kinds of organic calcium minerals precipitating in a lot of inside our bodies. And this one here, even in tumors, there's a lot of calcium, zinc, and iron. These calcium, zinc, and iron oxides, phosphate, and sometimes carbonates. And indeed, but carbonates don't stay there very long, and especially if you have certain special environments like pH is not so acidic. And when we have a surgery, for example, and some people use a bioactive glass surface for bone regeneration, okay, to facilitate regeneration like this over here. And so these serve as a template to nucleate and to promote regrowth of bone. And so these scaffolds are used as nucleating templates. And interestingly, some minerals can be eaten directly as medicine. This goes back to thousands of years ago in traditional Chinese medicine. And so, for example, these three here, you see all containing arsenic, right? Because arsenic is such a toxic metal. So if there's a lot of arsenic in water, drinking water is very low levels. It's toxic. But when arsenic forms sulfides and oxides in solid, these things here can be swallowed. Just swallow these things actually help. And one of the reasons actually just discovered in 2010, this paper is quite interesting paper. They say this particular arsenic oxide basically kills or suppresses certain protein, and otherwise the protein promotes cancer development. Okay, so basically it's killing bad protein that is responsible for the development of tumor. And so that's one needle in a haystack. Because going back to almost 2,000 years ago, a very famous Chinese doctor, and he did not have formal medical training, but he was just doing all that, and just survey hundreds of thousands of medicine, and including a lot of plants, a lot of minerals, and just test on his own body first to see what happens. And he has books like this. And so even up to today, nobody really knows what happened and what are the reasons why these minerals can actually be used as medicine. So that's a little preface of this medical geology and a little history. Now let's see a specific example. So there's a field I've been working on for the last several years, and how actually clays have a similar property that can be used for medical purpose. So first of all, let's define clays and clay minerals. Clays are basically a physical term. Anything that's smaller than 2 micron in size, we can call that material as clay. Okay, no matter what composition is. If it's smaller than 2 micron, 1 micron is 10 to the negative 6 meter. Okay, so 1 micron is a very small unit, and you cannot see this really with naked eye. And then if this clay less than 2 micron has silicate in composition, okay, has basic framework of SiO4 sheets, and then you have a whole bunch of other metals in between, then that's called a clay mineral. Okay, so a journal called Clays and Clay Minerals, basically including anything that's smaller than 2 micron. But a lot of that papers in the journal is about clay minerals, because these groups over here are very abundant and very common, and other ones are less common. So the 2 most abundant groups of minerals are silicates and oxides. And we find these very commonly in soils and sediments, and in the rivers and oceans. So
what's the magnification of that picture?
This one here,
yeah.
a few microns. I forget to put the scale bar. Yeah. So if you take any sample with a scope and analyze this, then most likely more than 50% of our soil is made of these clays. And mostly silicates, but some oxides. Okay? And for silicates, they have this layered structure here. And because it's internal structure, all the atoms are arranged in sheets like this, in layers. That's why physically, if they just keep growing big enough, you can see them with your eye. They occur in this kind of sheets. But if you look down from top, then they look like this. And these clays are used a lot, right? And in nuclear repository, in our household, okay? And also we eat these clays just like we eat arsenic oxides. Okay? And these clays are used for cat litter because these clays have a lot of space between sheets, between individual sheets. And this space can hold a lot of water, a lot of nutrients, a lot of anions, a lot of cations, a lot of organics, lots of stuff. It's almost like storage space for a lot of things. And so I want to talk about specifically about medicinal use of clays. Okay? And so if people eat clays, and for the reasons that, clays have high heat capacity. Okay, basically they absorb heat. And so that allows topical applications to stimulate blood circulation. So if we apply clay, right, and then heat is better protected, does not get dissipated easily. So then this area is warmed up and helps blood circulation. Also exchange of cations through skin pores and hair. And so because these clays contain a lot of cations in this area over here, these cations can be slowly released and exchange with toxins in our skin, then get rid of a lot of toxins from our skin. And also tubular minerals, clays, are used for absorption of excess fluid. And so that basically prevents swelling. And so a lot of things, for example, kaolinite and smectite and a lot of things are consumed, basically edible clays. And then obviously antibacterial, that will be the focus of today's talk. So we use clays in our daily life. So I don't know if anybody has this kind of experience. If you go see a dentist, what happens to your teeth if you have a cavity, or if you want to have a very shiny and polished surface? Actually, they give you. Anybody experienced on this? Nobody? You can ask next time. Ask your dentist. So this looks very shiny and very nice, right? So basically, a set of porcelain teeth that's made from clay, it's beneficial to health. Not only looks beautiful, right, when you talk, when you open your mouth, but actually it just makes this one here sturdy, protective, less decay, right? And then, but if you use the same kind of porcelain for this pipe, what is this pipe? Like smoke, right? And some people use this for hot water for making tea. Actually, could have both beneficial and harmful effects. This one here in particular actually develops some cancer because it has a lot of toxic metals in it, in this clay surface. But you say, why not on this one, right? And it's just different kinds of clays and different treatment procedures. And so you have to be careful if you want to use this for these kind of applications. And so a lot of people actually ingest clays directly. And for example, this one here, red clay, we call it termite mound. And this one here is red clay, is commonly ingested by women after childbirth and during monthly periods. And a lot of women, especially in Africa, tend to consume clay right here on the top. Because say this clay on the top is oxidized clay. Remember from last time, oxidized clay is healthy and has better purity than the ones down below here. The heavy ones and is heavy metals after multiple rainfall maybe reach down here, and so the ones on the top, remaining here on the top, better quality. They're trying to grab that and for ingestion. But if somebody eats too much clay in their lifetime, then you can have some blockage here. Intestinal blockage. That's not good. Okay, and so it's. But if you eat some, it's okay. It's beneficial. But if you eat too much your whole lifetime, that's bad. And actually, so now I want to talk about this antibacterial property of certain clays. And last time I didn't have time to fully develop this talk, but this time here we can go a little longer, so I have more time. So actually, this antibacterial clays was first discovered by a French woman, that's her name, and we call her Hailiang for short. And she was on a diplomatic mission with her husband to Ivory Coast, right here in West Africa. Right? And so in that region, a lot of people having this disease called Brody ulcer, right? It's a flesh-eating mycobacterial infection. And she bought two kinds of clays from France. And these are the names of the companies where she bought these clays from. Turns out these two clays look identical. Chemistry-wise, look identical. In mineralogy, they are made of the same kind of material, but one is not antibacterial, one is antibacterial. That mystery really triggered more than a decade of research after this. So this was about 15 years ago, 15 to 20 years ago when she first discovered this. And so this is initial, I had many more horrible pictures than this, but I thought right after this we're going to eat dinner, and so hopefully, right, you don't want to see a lot of nasty pictures. And then this is after a day, and this is after 3 months, and just using this natural clay from this one over here, and it helps and it heals. And very simple procedure, right? You just mix this with water and just pour onto whatever your infected area is, and over time it just heals. So if you look at the composition of these two clays, and we have all this, so if you're not a geologist, don't bother with this, but these are the individual minerals that make up this clay, two clay deposits. One, remember this one here is antibacterial, this one is not. So the dark is this one here, and the light is this one. And if you compare, it really looks the same in terms of the composition of what minerals make these two clays. And then Linda Williams is really a pioneer on this from Arizona State University, and so she was so curious at the time. She called me up, said, "Hey, are you interested in this? Let's work together." And I was just settled in Miami more than 15 years ago. I was busy enough to create my own lab and my group, and occupied with lots of other stuff. So I just dropped the ball, but she really moved ahead. And so she did a whole bunch of antibacterial testing here, but after almost 10, 15 years now, I came back to this again, and I discovered this is so interesting. There's so much more to do about 5 years ago, and we're going to continue on beyond this. So she did a whole bunch of antibacterial assays, and so both from, so she isolated bacteria from these clays, and she also isolated clays from these deposits. So clays and bacteria. Then she did a whole bunch of testing, both for physical interaction between these two and a chemical interaction between these two. Okay? But physical interaction basically, how clays and bacteria are interacting with each other physically. Perhaps some of the clays, they occur as like very sharp needles and the sharp edges, maybe just kind of just cutting and just penetrating into a bacterial cell, maybe physically killing that. And but chemically, maybe these clays actually releasing these clays maybe removing some toxins from this bacteria, or maybe if these clays are removing some nutrients that are required by bacteria. So if these clays are removing somebody's meal, right, and then they have nothing to eat, and they will be starved to death. Okay, and so these are possible hypotheses. And so to test the physical interaction, what we do is you put clay suspension inside this called dialysis membrane. So this membrane has very, very small pore size, like less than 0.2 or less than 0.2 micron. Okay. So clay particles cannot diffuse out, but anything that's soluble, if there are any metals released from the clay, they can come out and then interact with bacteria outside. So the clay is inside, bacteria outside, so there's no physical contact, but there's chemical contact because some anions and cations, metals released from clays can diffuse out of this membrane and then still interact with the bacteria outside. A lot of times what we do now, we do the opposite. We put bacteria inside, the clay suspension outside, because bacteria size is more uniform. Clay suspension is not so uniform, so always tiny clay particles leak out. But if we put a bacteria inside this membrane, bacteria 1 or 2 microns, they stay there forever. And then if we put clays outside, but again, clays can diffuse inward. So no matter what you do, maybe there's some physical contact. But the idea is to get rid of physical contact to see if chemical contact kills bacteria. And in this case, this is before experiment, this is afterwards. You can see all the red, green ones over here are alive, the red ones are dead. And you can see here, you don't see any green ones after the interaction. So it looks like the clays are releasing some toxic metals that may be killing the bacteria even outside this membrane. Okay, and so then Williams has looked at the size effect, and so and we have done the same thing here. So this is antibacterial clay, right? And so for bigger particles like this one here, it is the same as control. It's not antibacterial. But if you decrease the size to 1 to less than 0.2 micron, all of a sudden this clay becomes antibacterial. So the small size means bigger surface area, big exposure, right, to bacteria. So bacteria have more chance to interact with small particle size of clays. And these clays are more effective killing bacteria with smaller size. Then chemical interactions are more complicated. Okay, so these clays can release a lot of cytotoxic metals like aluminum 3+ and iron 2+, and also these clays can actually produce a lot of oxidative stress, and to oxidize lipids and proteins, and to increase membrane permeability, and but clays can also sequester nutrients that are required for the growth of bacteria. So somebody is enjoying a meal and all of a sudden you take the meal away and then they'll be starved to death. And aluminum binding to phospholipids leads to osmotic imbalance in the cell lysis. And so aluminum can bind to phospholipids. Phospholipids are on the outside of the cell wall, right? And so if aluminum binds to that, then it causes osmotic imbalance. So basically you have to have a balance in the salt content inside and outside of a cell. So if that happens, then it's out of balance. And then also direct formation of hydroxyl radicals from reduced clay, and I will talk about this more. And then also the pH effect and oxidation state. So some clays have pH buffering capacity that's beneficial, but some clays can actually produce a lot of acidic pH that will be antibacterial because a lot of bacteria are neutrophilic. They have to have neutral pH to make living. So and now we have tested all kinds of bacteria, gram-positive, gram-negative, and if you're not a biologist, give you a little basic knowledge. So gram-positive bacteria, this one here, you can see the cell wall is thicker. Okay, so this particular bond we call peptidoglycan is very thick, but gram-negative ones are very thin. This one here, but instead, this gram-negative bacteria have double membrane, inner and outer. So if these clays want to physically penetrate, they have to go through double door, like this, double door, right? And So you can see here the double membranes over here. And all bacteria, all actually microbes have gram-positive, gram-negative, just like all people have male and female. Same kind of idea. And then mycobacteria may have this little more complex surface structure here. So this is a typical antibacterial testing assay. And so this is the control, right? And then you test on different kinds of clays. So you put a very thin film of clay on this petri dish. Then you do the plating, typical, very simple microbiology, and undergrad students will do this in their first year in the lab. And they spread this little clay bacteria suspension. Then if you see no growth after overnight, then you know this particular very thin clay film underneath is killing all this bacteria. There's no growth overnight. But this one here, the control, you see very nice growth and these colonies. And another way to do this is actually we put let's see, bacteria first, and then you put some clays here, and then you see if clays are inhibiting bacterial growth, then there's a particular circle. Like so the bigger this diameter is, the more effective this clay clay is in killing bacteria. So if you put just like one single drop of clay in the middle of this petri dish, and if it kills all, that clay must be extremely powerful, right? Different ways, different relations of doing this kind of testing. And so now, actually, again, Linda has done most of this work, and she has surveyed 50 clays worldwide, and only 10% are antibacterial. Only 10%. I think and that's only like 5 clays, right? So if we want to do this commercially, obviously this 10% of clays can be quickly exhausted. And so this two particular clays from US, one is from Oregon, the other one from Nevada, are very effective. These two from France, remember this one here is very effective, this one is not effective. So she did all this testing over here, and obviously just so they say this gram-negative, gram-positive. Usually you can see the gram-positive ones, even though they have a single membrane but they have a thicker cell wall, it's more difficult to kill, right? And so for the same kind of condition here, you know, maybe they decrease by a few orders of magnitude, but for gram-negative ones, it's totally killed, 100%. And so these two are from US, Oregon and Nevada, and similar. And so what is the mechanism? So why is this 10% of clays are antibacterial and the other ones are not? And the reason is, well, it turns out in this particular plot, I'm plotting EH versus pH. pH here, the EH is a reflection of how oxidizing this environment is. So the ones in this, the black ones here, this one here, are antibacterial. And the ones here are not. And you conclude from this plot, well, if you have neutral pH, they are not bacteria, but if this clays, if you just put them into water, if the pH, natural pH is acidic or alkaline, they are antibacterial. It turns out these two fields are what we call stability field to solubilize aluminum. Like anything has a stability field, even for a person, right? Like tonight, you guys here, your most stable place is in this lecture hall, right? And for any material, for any mineral rock, they always have a stability field. Under a given set of environmental conditions, they find some combination to be more stable, right? So aluminum 3+ and in this particular case, most stable in aqueous under this kind of pH. So perhaps that means aluminum 3+ in aqueous solution is really causing the antibacterial activity. This one here is solubilized because at neutral pH, this one here precipitates as a solid. Obviously, solid precipitates. If bacteria in aqueous suspension, they have no chance to interact. But this aluminum 3+ is soluble, then they can kill bacteria. And in indeed, so if we do again EHPH and put all the aqueous speciations, aluminum iron, indeed, in this particular acidic pH, iron 2 is soluble. And in either acidic or alkaline pH over here, it's out of the plot, aluminum 3 plus is soluble. So maybe soluble Fe2 plus and aluminum 3 plus are the reasons for antibacterial activity. Okay, and so the reason is this ferrous ion, okay, aluminum 3 plus, I already talked about toxicity because aluminum 3 plus binds with phospholipid, causes osmotic imbalance. What about this iron 2? This iron 2 turns out goes into the cell and it triggers all these reactions over here. So this is M representing metal, but you can just replace this with iron 2. This iron 2 reacts with oxygen to produce superoxide radical, hydrogen peroxide radical, hydroxyl radical. So these radicals are strongly oxidizing, right? They can oxidize proteins, they can oxidize lipids, basically break down proteins and DNA. And so the iron 2 goes into the cell and then triggers all this reaction here, then iron 2 is oxidized to form oxide. It's a solid. So you can see right here. Actually, you see under electron microscope image, you can see precipitation of this iron solid. Then, if you do a chemical compositional mapping, so inside, green stuff here is indeed Fe2 plus. This is why Fe2 plus goes into the cell, triggers all this we call Franklin reaction, right? And then precipitating is a solid. But outside, the red one here is Fe3 plus. So it is Fe2 plus that goes into the cell and it triggers all these reactions over here to produce these radicals. These radicals are killing the bacteria. And then aluminum, for example, here, you actually see the aluminum on this outside, right on the edge. So the toxicity of aluminum comes from binding of aluminum to phospholipid on the cell membrane right here. But inside, you see a lot of iron, which is mostly iron 2. Okay, and so now that's the overview of the current state of knowledge in this field. And then remaining stuff is really what my group has been working on, and I touched a lot of this stuff in the other talk. I'll go very quick. I'm trying to finish this in about 10 minutes. Is that right? All right, Chris?
Yeah.
So now, as I said, there's only 10% of all clays, 50, right? Only 10% is only 5 out of 50 are antibacterial. So now really the tricky, most important question is, can we make just normal clay, not antibacterial, into antibacterial, right? And most importantly, so the stuff I talked about so far, this requires soluble metal. And metals don't get solubilized under neutral pH. Metals get solubilized under acidic pH or alkaline pH, right? This one here, either very acidic pH or alkaline pH. But these pH conditions are not friendly to human skin, right? Sometimes in early spring when kids play soccer in the field, right? Especially in industrial city like Cincinnati, which is very close to Oxford. And their back, their face, after soccer game, right? My kids used to play that when they were small. All becomes so red. You guys know why? Acidic rain, right? pH is like 4. 4 or 5 is very common, right? And not all the time, sometimes. And so obviously that's not skin-friendly pH to have solubilized metals like this for them to work. So really our contribution here comes in, can we make normal clay antibacterial under neutral pH? pH 6, 7, 8. That's user-friendly, skin-friendly pH. That's really the key question number 1 here. So, we have tons of clay deposits, and we're like a Wyoming bentonite. And if we can make these clays antibacterial, that will be unlimited supply if we someday commercialize this. And so, luckily, this clay is always to a different amount, always contains certain amount of iron in the structure. And I mentioned this already, this iron in the structure can be reduced, so mostly the clays contain ferric iron if it's repressed. But we can reduce it to ferrous chemically or biologically, and then this Fe2 plus reacts with oxygen in air to produce all these radicals. Now remember the difference? So the earlier I'm talking about this Fenton reaction here we call homogeneous, because this Fe is insoluble in aqueous in water. Okay, but here I'm saying these kind of reactions can occur in solid, right? As long as you have aqueous ion 2, if you have any solid ion 2, you can have this kind of Fenton reaction. This is called heterogeneous reaction, right? Not homogeneous in aqueous solution. So now you produce all kinds of reactive oxygen species, including iron 4, which this is kind of weird oxidation state for iron, and then go through the same kind of reactions, kill Fenton reactions, and so these radicals are very effective killing bacteria. Without going into a lot of data slides, I'm going to skip all that stuff. And the basic idea is that we have done my first PhD student, when he was studying on this, always every single day he was counting hundreds of petri dishes, just visibly counting, oh, is there any growth over here or is there any killing? He was hoping not to see anything overnight, right? But overnight, in the incubator, if you see this kind of stuff, that's bad news, right? Not killing. And it's so very simple. So what we did is we did an experimental group with reduced clay in lots of air in the headspace, and then this is the control group, right? And this is unreduced clay, just oxidized clay, and then you do this for a few hours, and then you take suspension, the cell suspension, and put on the petri dish to see how much killing has occurred. And so we have verified these reactions over here. If we remove oxygen, what happens? If we remove oxygen, none of this will be produced. They're not antibacterial, right? Then that's basically inside a grow box where there's no oxygen. Then we can vary different amount of iron 2 and under different pH conditions and all different combinations. And also we were able to show this clays can be recycled multiple times. So if you use the same amount of clay and then you reduce it, then you produce all these radicals, they kill bacteria. And then after they get oxidized in air, this will be oxidized to ferric. But we can reduce this ferric back to ferrous because clays don't dissolve. They're still there. After your first treatment, you can actually recover that clay. And the clays are so cheap. But if some clays are so precious, you want to recycle, reuse it. Almost like reuse needles in hospital, right? That's in old days. These days, you don't reuse needles. But who wants to reuse this clay, right? If this clay have been used in somebody else's skin. But the idea, in case, like for example, this is a French clay deposit. The first one is the effective one has been exhausted, right? So it would be nice to reuse that. It's especially on the same person, right? On your own skin, they reuse it, right? And so that should be fine. So it turns out we have done 3-5 cycles, and it works same effectiveness, same efficiency. And then we have visually observed how this killing occurred. Turns out here, this one here, the cell membrane has a lot of complex structure, lipids and proteins. And for this particular one here, this one is on E. coli. Okay, you get diarrhea, more than likely you have E. coli, right, infection. And so that E. coli is a pathogenic, some, not all. And so it turns out if you visually observe this, after the clay interaction, you see their ends, okay, on the heads over here. Some part here is slowly falling off. And we watch this over time, and it's this, and then starting this, and then gradually big cracking open. And that's because the phospholipids are getting oxidized by radicals from these oxidation reactions. Then we can actually visualize, we can see what's happening inside. We're using this called DCFH-DA. This way here is a probe, and basically it gives different kinds of color. We call fluorescence color. But if you have these radicals will quench this fluorescence, then Actually, now this particular agent does not give any color. But if you have reactive oxygen species reacting with this, then they give fluorescence color. Green and red, whatever color you want to see. At the same time, we use a different probe to see where's the iron, intracellular iron. And it turns out this one here gives fluorescence without binding with iron. But if there's iron inside the cell, then this binding of iron with this probe will quench this fluorescence, and then you don't see any color if there's iron. So this one here, the two ends basically have no fluorescence, meaning there's a lot of iron on the two ends. And then you have a lot of reacting oxygen species on the two ends. So these are the locations where iron 2 goes in there, accumulates, produces all these reactive oxygen species. These reactive oxygen species are so strongly oxidizing, and they are basically oxidizing proteins, lipids, and then DNA, and then kill the bacteria. So this is the model, right? So you don't have to have acidic pH, more or less neutral pH, and then if we oxidize this iron 2 in the clay to oxygen, produce hydroxyl radical. This radical so strongly oxidizing, and then basically breaks up some membrane and makes this membrane more permeable, and then a certain amount of soluble in a tiny fraction, very, very small amount, goes in there and then triggers additional Franklin reactions. So it's a combination of this, the membrane oxidation, and then Franklin reaction inside the cell. It's a combination of both that kills bacteria. So more recently, we say this process is not so efficient because bacteria are negatively charged and clays are negatively charged. They tend to repel each other. So bacteria are way down here, and if you have a lot of, if you have radicals on the ceiling, they don't see each other. So only by random chance, maybe 1 out of 10 bacteria get killed. Okay, so now we want to maximize this efficiency by bringing these two things together. So we can do this by reversing the charge of the clay, so this clay becomes positive. The bacteria is still negative. So now we have electrostatic attraction. These bacteria are waiting there to be killed. Right? Nobody does that, but the bacteria are silly enough and they don't recognize, right? And there's actually lethal hydroxyl radical there wants to kill me, but still goes there because of electrostatic attraction. So we have done that stuff, and so this is the earlier model. The efficiency is not very high. You have to have a lot of clay each time to put on to kill. But now with this modified clay, we can actually increase the efficiency by a factor of 10 or even a factor of 100. So we use very little clay and it kills the same number of bacteria. Simply, we are reversing the charge of the clay from negative to positive by inserting a positive charge polymer, we call cardisan, into this. Then the whole surface charge of the clay is reversed. And this cardisan is biodegradable. It's not really toxic itself, and it's readily available, very cheap. So this modification is quite effective, and I'm going to skip a lot of data slides. And now, in this case, you're thinking this bacteria and clays are right next to each other spatially. So obviously, they see the killing, the membrane damage, not just on the polar region, but across the entire cell. And just as long as the bacteria and clay are in contact, so the point of contact is where the damage takes place. So if you have a longitudinal contact, then the membrane damage occurs throughout the entire cell. And similar kind of imaging techniques, that's the model. And lastly, just 1 to 2 more minutes. And so now we have solved the pH problem. We have made this clay more efficient. But lastly, people's skin always sweat. Right? There's a lot of all kinds of sodium, potassium, and salt, and also some oily stuff on our skin. Right? If I don't wash my hair, a day or two, my hair gets oily. Right? And so a lot of organics. And so we want to simulate the natural environment to see under skin environment, is this process efficient? So we are adding sodium chloride, it's just salt, or calcium chloride. Turns out if you add calcium chloride or sodium chloride, they're no longer antibacterial. So if you don't have any salt, they're very efficient removing bacterial cell, decreasing from 10 to the 8th, 10 to the 4th. You kill by 4 or 5 orders of magnitude. Sometimes you just killed all 100%. But if you gradually increase sodium chloride, little salty environment, then they survive. And the calcium is more effective than sodium. And and also lactate and oxalate and citrate, right? And this organic molecules, citrate is most effective protecting this cell from being killed. And so if you don't have any of these organic ligands, then you kill a lot. But if you add these, like especially citrate, it's a very common small organic molecule, then this cell is protected. So now actually Gary has a lot of mutants and has a lot of chelators. We're working on this for some more and trying to understand about chemical mechanisms and to really simulate this environment much more realistically, and also test on animals next. And with that, I think I'll stop in the time, maybe a few more minutes left for questions. I don't know if you guys have any. Thanks very much.
We need to talk about so forward to questions. Can you excuse my ignorance? This is not my subject. First time I hear about it. not here, of course. I look at what I can. Can you synthesize clays in the lab?
Yes.
So, if you can synthesize clay in the lab, why did you say only having 10 of those effective clays will be adequate?
Yeah, clay synthesis is difficult because it requires very high temperature. So it's costly. And you have to have a special apparatus. So some people do. And if you have to have very 100% purity and specific chemistry, you can do that. And also it's a slow. You cannot really synthesize a huge deposit of clay. That's almost like impossible. Only like a very small amount in a lab. Yeah. Yeah.
If you could understand the process by which the bacterial properties work, could you just forget about the clay and just incorporate those in some sort of other carrier?
Put this radicals into some sort of carrier? Yeah. The reason is clays, in addition to these radicals, they also provide nutrients. Actually, all these other things I mentioned earlier. But some people put these, not really this reactive oxygen species, some other like drugs onto some other carrier, some more like a synthetic material for very slow release. That's another medical geology problem. Some people actually use clays to put like drugs into the interlayer and then have very slow release. Because if you just eat drug directly, then just quickly the effect goes away in a day or two. But if you embed drugs onto clay or in the clay, then maybe the effect can last for weeks. And so this is used in animal feed, for example. Like in big farms with pigs, you put antibiotics and embed into clay structure and then feed them, and then they're going to kind of just get this antibiotic slowly, multiple weeks. Yeah.
How long does clay stay in the digestive system?
How does it stay?
In the digestive system, if you eat it.
Yeah, the hope is that they absorb, they absorb a lot of toxins in the metals and the water, and hopefully just come out. But some people, they say, if some people eat too much clay and they stay in the intestine, they get clogged. That would be a bad effect. Then you have to have a surgery and remove that physically, almost like kidney stone, So, yeah.
Do bacteria naturally have hydrogen peroxide in them anyway from other oxidative reactions? So you can kind of cut out the first two parts of the Fenton chemistry and go straight to the hydrogen peroxide?
Exactly. So some bacteria, just from their basic respiration, they produce hydrogen peroxide.
Like we do in our lungs.
Yeah. But the amount is not enough to produce as much radical. So some people have done, actually we have done that too. So if you have just from microbial respiration, you produce some hydrogen peroxide, but these reactions cannot be sustained if you just use that hydrogen peroxide from bacterial respiration. Yeah. Okay.
And to follow on, do you need the clays at all? Can you not just give people iron and aluminium, and it sort of follows off from some other people's questions? So if you just give aluminium and iron, if they're the reactive components. So if it's a wooden iron and aluminium, why do you do clay? Because it's a leaching out process, isn't it?
Yeah, it's but here's the trick. So if you put soluble iron, right, and they can diffuse, not only kill bacteria, but maybe diffuse and keep going into your skin cell. You never know. Yeah. It's difficult to control. But the clay is, much bigger in particle size than ions. So physically, it's a barrier. They will not go in. They will not diffuse in there. And also, in order for iron and aluminum to be soluble, right, you have to have acidic pH. Otherwise, if you have neutral pH, you can never maintain these metals in solution. And they will be oxidized anyway. So if you have neutral pH 6 to 8, you'll form iron oxides and aluminum oxides. These oxides are not effectively producing radicals. So if you have these oxides in air, there's no radical production.
And how do you achieve selectivity for the bacteria? Because reactive oxygen species will damage human cells as well.
The hope is that we can specifically modify the clay surface so we can control where the hydroxyl radical is produced. So, the clay structure has layered structure, so we can say, radicals should be produced on here, on the basal plane, on the edge, like this specific direction, and perhaps, but that will be a future direction. We don't really know for sure at this point. It will be nice.
But the clays did work against Burley ulcer.
Yeah.
And they healed it.
Yeah.
And they obviously were killing bacteria rather than the human tissue.
Right. So we have worked on, we have used this clays for like a human cell, eukaryotic cell, fungi, and they don't work on fungi at all. They don't kill fungi at all. Because the human cells or eukaryotic cells in general have very different cell wall structure, right? And their DNA stuff, mostly they have multiple protections. So that's the hope. Just by nature, they don't work on human cell, and they kill bacteria, prokaryotic cells. Yeah. So that's the hope. But evidence so far suggests that way. Yeah.
If I could just ask a final one, I was interested when you went down your particle sizes, it didn't look just like a normal particle size effect. It was like a step change. It went from
Yeah, right.
So you would expect to see, if it's diffusion limited, you would expect to see some change, some continual change, rather than a sudden switching on of that.
Yeah, just like actually, I think, I don't know if from Linda's work, from somebody else's work, and we have done a similar thing, but we don't have enough data to show a trend. Yeah, it's a little strange, isn't it? There's nothing, no killing, all of a sudden 100%. There's a big jump.
Almost like it's become ingested or something, rather than it comes to the side where it has to be.
Could be. My guess is there's a big jump in the particle size from this to this. So, but again, this is less than 0.2, this is 0.2 to 1. Yeah, so it looks like continuous.
Okay. Well, in that case, our IS fellows are also hosted by our wonderful college systems. In this case, Professor Dong is hosted by Joseph Butler College. It would normally fall on the master liver of voter facts at the end of this lecture, but I'm standing in as said master today. So we can thank Hailiang again, and we hope you enjoyed your evening. See you again at the next IAS talk. Thank you.